Welcome to this lesson on bacterial reproduction. Today, we'll explore binary fission, which is the primary way bacteria reproduce. Binary fission is a fascinating process that allows single-celled organisms to multiply rapidly and efficiently. Binary fission is a form of asexual reproduction where a single bacterial cell divides into two identical daughter cells. The term asexual means that only one parent is involved in the process, and no genetic material is exchanged between different organisms. This is the simplest and most common method of reproduction in bacteria. Binary fission has four key characteristics that make it unique. First, a single parent cell produces two identical offspring with the same genetic information. Second, it is the most common form of bacterial reproduction in nature. Third, the process is remarkably simple and efficient, requiring minimal cellular machinery. Fourth, it allows for rapid population growth, which is why bacterial infections can spread so quickly. The binary fission process occurs in four main steps. First, DNA replication begins, where the bacterial chromosome makes a copy of itself. Second, the cell elongates and the DNA copies separate, moving to opposite ends of the cell. Third, the cell wall and membrane begin to pinch inward at the center, creating a division point. Finally, two identical daughter cells are formed, each containing a complete copy of the genetic material. The first critical step in binary fission is DNA replication. Bacteria have a circular chromosome, which is different from the linear chromosomes found in human cells. During replication, this circular chromosome duplicates itself, creating two identical copies of all the genetic material. This ensures that each daughter cell will receive complete genetic instructions. One chromosome becomes two, and this happens before the cell physically divides. After DNA replication, the cell begins to grow longer through a process called elongation. As the cell stretches, the two copies of DNA move to opposite ends of the cell. This movement is guided by special proteins that act like molecular motors, ensuring each DNA copy reaches the correct location. This separation is crucial because it guarantees that each new cell will receive exactly one complete set of genetic information. The final stage is called cytokinesis, which is the actual physical division of the cell. The cell membrane and cell wall pinch inward at the center of the cell, similar to tightening a belt around your waist. This pinching continues until the cell is completely divided into two separate cells. The result is two genetically identical daughter cells, each capable of growing and dividing again. This completes one cycle of binary fission. Binary fission is important for several reasons. First, it's incredibly fast. Under ideal conditions, some bacteria can divide every 20 minutes, meaning one cell could theoretically become millions in just a few hours. Second, it's highly efficient, requiring minimal energy and resources compared to other forms of reproduction. Third, it enables exponential growth, where the population doubles with each generation. Finally, it's reliable because it produces identical genetic copies, ensuring the offspring have the same successful traits as the parent. This combination of speed, efficiency, and reliability makes binary fission one of nature's most effective reproduction strategies.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.